I hope it's not too ambitious. But I'd like to start by thanking the organisers, both for letting me talk here today and for inviting me to spend so much time with some great people and learn so much. So by the end of the talk, I hope this title makes a bit more sense. So this is my introduction, which is basically we do some modelling of sequence evolution. And the most basic simple models we think of make a series of assumptions. And so we have our model here, we have our rates of change between our different nucleotides, we have a tree, we have a sequence alignment, and we assume that all the sites in our alignment are evolving to the same process. We also assume that all parts of the tree evolve to the same process. And we've heard some good talks this morning, for example by Andrew, showing that you can allow different sites in a sequence to evolve differently, and it provides an important description of how evolution takes place. We've also heard from Tanya that there's various complexities of interactions between those sites, and that can manifest itself in a variety of ways. So I'm going to start off talking about spatial heterogeneity in sequence evolution. So this is the idea that evolution is a heterogeneous process. For this example, let's say we've got 3 different models: a fairly slow model, a medium rate model, and a faster model. And each one of these not only varies in rate, but can also vary in the kind of composition of model you have. So this one has equal nucleotide frequencies with a bit of transition-transversion ratio. This one tends towards, I believe, C and G. This one tends a bit more towards A and T. So spatial variation is that we pick a site and it evolves to a single process. And the thing to note is that the tree remains exactly the same. It's just a scaled version, and all the way through the tree it's evolving to this type of process. This is in contrast to, say, the ideas of Pagel and Mead, where not only do you have a different process, you have different branch lengths. So when I talk about spatial heterogeneity, it's only among sites in the sequence. Similarly, we have another site, evolves to another process going a bit faster, so the tree stretches and evolves in a different way. So another thing you can think of is temporal heterogeneity, so heterogeneity through time in evolution. So you can pick a site this time, and instead of saying that it evolves only to one of these models, we can say it evolves to a multiple set of these models. So for this part of the tree, in this case, it evolves to the slow model, and then for these 3 sequences, it evolves to a slightly faster model. Now, this can actually end up getting things that are quite complicated. For example, this tree here, where there's been a lot of switching between all these processes, and the switches are also occurring mid-branch, which is also worth noticing. And it just looks a mess, basically. It's very, very complicated, and it'd be difficult to build a simple model for this. So I should also say here that temporal heterogeneity has been investigated in some ways, but normally only as heterotachy. That's only what people have worried about. They haven't worried about the other variations in the model. So onto my motivation. So it comes both biologically and kind of from a more modelling perspective. We know that not including heterogeneity leads to inaccurate inferences of the tree topology and various other things. We've seen quite a few talks. So Gavin and Peter gave talks showing this problem. It's the actual general form of heterogeneity is fairly poorly characterised. We know there's heterotachy, but we don't know exactly what's causing it, what it is. So Andrew's talk, for example, gave a good example that protein structure influences it, but we're still some way of understanding exactly how it influences the pattern evolution. And understanding heterogeneity can lead to some biological insights. From a modelling perspective, we need to describe this kind of general heterogeneity. Tandy was saying that evolution is always more complicated than that. It's always more complicated than that. So from a modelling perspective, it's quite appealing. The model should also be identifiable, because otherwise there's not really much point having it if everything just comes out looking the same. And John and Elizabeth have shown that. These kind of models I'm going to talk about are going to be identifiable, and it should also be computationally efficient. So what I mean by that is we should have few parameters, we should have a smallish number of states. We don't want, if possible, the say 20 to the n by 20 to the n matrix. That's not really computationally easy, and ideally it should be applicable to tree search as well, because a lot of people are interested not because of the models but because of the trees. So I'm going to introduce an idea. These have been talked about before by various people, and I'm going to give them another name along to the long list of names they already have. And these are called temporal hidden Markov models. It's a general type of model. It can describe both temporal and spatial heterogeneity, but it has the advantage of allowing simple likelihood computation. And when I say simple, I mean it's you can describe it as a reversible process, as it's stationary, and it's IID, so you can break up the calculation. So these have only really been applied to rate heterogeneity. So the covariant model of Tuffley and Steele is an example of a temporal hidden Markov model, where the hidden state is that rate is varying. And there's a list of other names here, and John gave a more exhaustive list than this, but there's quite a lot more. So what do these things consist of? So they consist of a series of hidden states, and these are the substitution processes. And each of these hidden states describes the rates of change between observables. So in this case, we've got G hidden states, and the kth hidden state is defined by your rate matrix, and in this case, it's an HKY matrix. So it's HKY is completely general. We have a nucleotide distribution of hidden states. So that's the nucleotide frequencies we see. We have a rate. We have a transition transversion. There's no reason why you can't use this as a GTR. It just adds more parameters to model. So we have a series of these. Each of them can be completely independent from the other. So we have a set of these, and now we want to describe some kind of interactions between them, how they change into one another. And I'm going to describe that using a switching model. So this is just simply this is a model of what's happening at the hidden state level. So this is analogous to the rates of transition between hidden states in most other hidden Markov models as well. So this is defined by a G by G matrix, which is called C from the description of this type of model by Wang et al. So Ed Susko and Andrew Roger. And I varied it a little bit from what they've done. You can describe this rate matrix actually just as a kind of, almost as a GTR model among hidden states. So you have rates of transition, or exchangeabilities between different hidden states described by this rho. And you have the probabilities of the different states described by this pi tilde parameter here. And again, all of these can have various restrictions placed on them, and you can estimate them from your data. So to define the temporal hidden Markov model, you need to just combine them all together. And this gives you a bigger matrix. It's big, but not as big as it could be. So what we have is that we have parts of the model describing transition between observable states. That's our M's from before here. We have our parts of the model describing changes between hidden states. That's our C matrix. And you also, it turns out, need to include a factor from your models describing transitions between observable states to make it reversible. And when you build a model, it's fairly straightforward. The equilibrium distribution is simple. And the other thing to note is that hidden states and observable states do not change simultaneously. But that's the same as any hidden Markov model. You don't tend to get simultaneous change. So I like to think of these things as pictures because I can understand pictures. I still struggle with equations, being originally a biologist. So what we can kind of have here is that we might have a nucleotide in a sequence that starts off in our hidden state 1. It evolves a bit to hidden state 1 all the time. It's got a probability of jumping into one of these other hidden states, and that's defined by these things on the diagonal here. So we evolve along a bit, jump across, say, to state 3. It evolves along a bit faster now, jumps back again to, say, state 2, and evolves again. Now, this kind of describes the pattern of this description of temporal heterogeneity quite nicely. We have variation both amongst sites in the sequence because they're independent. It can take each site can take any of these forms at any point, and we also allows it to change during time as well. And there's a couple of other things you can show. So the C matrix, it's just a kind of summary of the kind of transitions that take place between the hidden states. And another interesting thing you can look at is the rate of transitions between the hidden states relative to the substitution rate. So you want this number to be pretty small, because if it's 2 or 3, we're jumping hidden states more than we're jumping observables, which are just, well, it's absurd. So one of the nice things about this is that you can start placing restrictions on the model, and you can reduce the model to just spatial heterogeneity. By setting all your row parameters to 0, you end up with a mixture model. So similar to what Andrew described this morning. And in this case, once a site starts in a state, it's stuck there, and it evolves the same all the way through time. And that's exactly the equivalent of breaking it up like this. That's what most people do with mixture models. This is just a very inefficient way of calculating it. So we've got a model. Have a look at some data. What can we find out about it? So this is some data picked from the literature. It's a GROEL gene, which is a chaperonin gene, and it's been used previously by Herbeck et al. To examine the origins of primary endosymbionts. Now, I can't comment exactly on why the biology is interesting in all this, but what they have shown is that GC content, for example, varies amongst the branches of the tree, and if you don't account for this, you get the wrong tree topology. So it seems like there's something going on here temporally. There's probably spatial variation that normally is in sequences when you've heard about rate. So it seems a good starting point for something to look at. And I should also note, just say, there's 23 sequences of and they're fairly long, so there's a fair bit of information there. So I'm going to investigate this in two separate parts. The first part I'm going to ask is, well, I've got a simple HKY model. That assumes everything's the same. I'm going to throw on top of that spatial heterogeneity, so that's variation along the sequence. How much improvement do I see by observing that? And I'm going to split that up and look at the contributions of adding rate variation amongst along spatial heterogeneity in nucleotide frequencies, and spatial heterogeneity in transition-transversion ratio. Then generalize the model a bit more and look at how important you see variation by allowing the temporal changes to take place. Just kind of quantifying what's there. It's which I believe no one's actually done in this form before. So the results come out in the table. So what we have here is the first thing to look at is that number there. That's the likelihood, log likelihood for under the HKY model. The number at the top here is the log likelihood improvement by adding a mixture model here with two states that allows rates to vary. And you get a very large improvement in likelihood. The number in parentheses is the improvement in the AIC over the HKY model, so an indication of model fit. In both cases, you're looking for bigger numbers better, basically. So what you find is you add rates, it's really important, you get a huge increase in likelihood. You add another hidden state describing more rates, you get a much smaller increase in likelihood, but still pretty big numbers. And interestingly, you have your spatial variation in rates. So this is a two-state. You add a parameter that allows switching during time, and you see a very, very small increase in likelihood. You don't see any importance for apparently from the data for switching in time, which seems a bit surprising and a bit disappointing. It's gone to all this elaborate effort to build up a model, and you don't see any improvement. Similarly, what happens is with nucleotides, improvement smaller, but I'm not going to talk about that too much. What I'm going to say is rate variation amongst sites is really, really important. If you don't include it in a model, you can't see anything else. And I'm going to pull out an example here, which is this model here, which is 3 hidden states where nucleotide frequencies are varying, and draw a picture of it. And the picture looks like that, which is a pretty unsatisfactory model. What it's saying is that there's a bit of nucleotide evolution here, a bit of nucleotide evolution here, but most of it, or well, a fair proportion of the time, I think it's about 65% of the time, it says if you're a G, you stay a G, which is a model of rate variation. It's invariant sites. It's nucleotide models trying to, with nucleotide frequencies varying spatially and temporally, trying to describe invariant sites. So that's not very useful. So what you can do instead is take these kind of big matrices and say, right, let's say there's rate variation across sites already. We've got standard methods of describing that. We've got a gamma distribution. So I take the whole matrix and say there's 4 discrete versions of it, each evolving to a different substitution rate. Does that allow me to now start learning a bit more about what's going on in the variation in nucleotides evolution? So this is the same data with a gamma distribution included. So do we find out more? Yes, quite a lot. We can already see that rates has dropped down to very relatively small values. It's still important, it seems, to include a bit of extra rate variation, but not a huge amount. And you can also see now that it's the improvement in likelihood obtained by adding temporal heterogeneity is improved and is now becoming much, much more significant. Similarly, with free state models for rates, it's the same again, and a much bigger improvement in improvement by describing temporal heterogeneity. Nucleotides, the improvements now are much smaller than before, because before they were trying to describe rate variation. We've got something that's doing that. But there's pretty big improvements, 200 likelihood improvements. That's quite large. People care about those kind of numbers. And there's temporal variation as well, which we can see, which we already knew from this data. And it looks like with nucleotides that you start adding more states, the improvement increases quite a bit more. And I can say if you go to 4 states, it increases again, but not by quite as much. But there's a reason why I'm not going to 4 states. And in this data, it looks like the most important thing, if you after including rates, is to allow your transition transversion ratio to vary a bit. And again, there appears to be variation in that. So this model in the bottom corner shows what happens with the with temporal variation and spatial variation in everything with the most complicated switching process you can have. And it's the best model. It provides the best fit for the data. And if you were going to use a model for inference, you would probably choose this one. So that's one data set, which is interesting, can learn something from that. What about looking at lots of data sets? What more can we learn? So I should also say, this is another kind of picture. Now you can see the nucleotide frequencies look like they fixed themselves somewhat. It's more like a kind of model you would expect to see, or would hope to see anyway. And this is another example. So more results. This is now from across 16 different data sets. And it's all just piled in together. There's a variety of different number of species in the data sets, ranging from about 20 to 100, and the variety of lengths and alignment length. And again, what we have is our improvement in likelihood is the top value, but that's just simply a sum across data sets, and the improvement in AIC, which is again a sum across data sets. You start quantifying the different types of variation you see again. Again, adding extra rate variation isn't that important, but allowing it to vary during time is important. Allowing frequencies to vary both spatially and temporally becomes very important. And in fact, if you're only going to choose to allow one thing to vary, this is the guy to go for. And again, kappa is important to include some spatial variation in kappa, and there is some temporal variation as well, but it tends to tail off as you add states. And note again, the most general model is the best in terms of fit. And for all 16 data sets, that's the case. The most general model, you need to include variation in everything. Everything varies. So, look at those data, and you can come away with a message, well, everything varies. It's useful. Yeah, great. So what can I do with it? What matters? Are there any relationships in the data that are interesting? So one of the things that seems to come out is that as your tree length grows, you seem to have more and more heterogeneity in the data. Now, I've plotted here just, this is for the most complicated model, the bottom right one. And tree length goes on. This is now AIC scaled for a number of sites. And there's a general pattern up. There seems to be actually 2 lines here, which I don't understand. There might be something interesting there. I'm not going to argue about that, but I'm going to say it goes up. So more evolution, more heterogeneity. And here's some more graphs that show the same thing, basically, for breaking it down however you want to break it down. And I think this is an observation, interesting observation. So, and what can be causing this? Well, there's something wrong with statistics is the first thing. So if there is, I'm sorry, and this next bit of the talk is going to be really boring if someone points this out at the end. But it could be something biologically interesting. And what could it be? Well, we start off with a sequence. We've got some variation across it. And we evolve it for a little bit. A little bit of time takes place. Now, a few changes have taken place. We know there's some heterogeneity in the data, but we don't know. It's because we've made very few observations, we can't say very much about it. Evolve it a little bit longer. There's some heterogeneity in the data. There's very few of these switches between the different colors. So spatial variation would seem to fit that quite well. It's few changes between hidden states, lots of things staying the same state. Great, that's a good description. As it gets longer still, more and more switching is taking place. So we can no longer describe it just simply by spatial variation. You need to describe it by temporal variation as well. Right, now on to the title of the talk. Simple model for describing complexity. So what kind of things can cause this variation? Well, the degeneracy of the genetic code is one obvious one. If we consider the third position in a codon, what we've got are varying degrees of degeneracy. So we've got our fourfold degenerate sites, which are evolving fast. We've got our twofold degenerate sites, which are evolving kind of medium speed with some kind of different pattern. We've got our slowly evolving sites that are one-fold degenerate that don't change very much at all. So there's some structure there, and this kind of model describes it by saying, right, there's a hidden biological process that's going on that's the first two codon positions. We don't know what that is. Let's just kind of have a series of hidden states that are changing between each other to describe it instead. So this kind of heterogeneity is going to take place in all nucleotide data you examine, and we just ignore it entirely. This is a form of heterotachy that people just haven't seemed to talk about too much. You sometimes just ignore the third position, but not try and describe it. And there are other types of complexity that fit this kind of description as well. And one description would be any sequence where your biological function places restrictions on how your sites change, and those restrictions have the potential to vary over time. You would expect to see this kind of process. And to me, I think that definition is basically evolution. You have a sequence, you've got a functional unit, there's restrictions on it. That functional unit is going to be changing over time. Those restrictions are going to be changing. So a hidden Markov model might be a way of describing that in a very general, simple way. And some conclusions, which I seem to have been overrunning quite a lot, so I will very quickly go through those. You've heard temporal and spatial heterogeneity. It occurs everywhere, although rate variation masks other effects. Most complex model describes everything best. Adding more states to the model improves fit as well. Biological causes of heterogeneity. Well, it's going to occur whenever there's biological function, as far as I can work out. I'd like to hear a counterexample if you can. Long evolutionary times may require even more sophisticated models, so more states, maybe even greater degrees of sophistication to the model. But if we're going to be getting back into these really, really deep relationships, we're going to need very complicated models. And temporal HMMs perhaps could provide a framework of doing these inferences, and perhaps we can also use them as a simple framework of investigating other dependencies in the data, rather than having to go straight for the most complicated model first. So once we've found some dependencies, then start tearing them apart biologically using these really sophisticated models, say of energy and so on in proteins. Okay, thank you.
Yes, two.
Yep.
The first is about the computational results.
Mm-hmm.
That is quite technical. Did you use the model to simulate the gamma distribution like the first presentation, or for example by substituting the mode or the transition probability matrix with the moment of the generating function of the gamma distribution?
So the way the gamma distribution is included is that you have a single parameter describing rate variation,
okay.
And you discretize it into your gamma distribution into 4 categories, and a value of each of those categories is then used to describe the overall
then
substitution
you
rate.
just insert it in the time. You make time.
Effectively, it's multiplying by time. Yeah.
Okay. And another question is, what do you think if you take, for example, the Kolmogorov equation, you imagine that R is dependent by T. Mm-hmm. Well, normally we take P dot of T equal R times P of T. And R is considered typically constant. But there is also a generalization of this model in which you can take R depending by the time, mm-hmm. which is a mess in the general case. But I can assume that the function of R are piecewise linear. And for example, similar to to match your model, so you block the state that you have indicated. Do you think that this kind of model could be more suited or similar, or could lead to similar results of you, or is worse, is better,
So in terms of data was appearing, series of observations were made according to the fact that you've kind of got blockwise.
That, for example, you can consider that in a given interval, for a given interval, the rate is constant. So, mm-hmm. for example, if I'm observing that a given subblock, I have a taxon a subset of taxa, and they have in the same position the same literals, for example, T. So I can say that more or less in this part of the tree, this is more or less constant. But then I have another subset in which I have a lot of change.
Yep.
So I can imagine that after a given instant of time, I switch from a rate to another level of a rate. So I have a piecewise linear function.
Yep.
With the jump that are equivalent to the states that you are considering.
Yep.
In this case, the integral of the Kolmogorov is different from respect to just the classical equation we are using. And so, do you think that this type of model could be interesting to use, or not really? Or
so in terms of when you've got it's a non-homogeneous process, is what you're kind of saying
yes.
in some way. So it's this model. Yes, that's the part of the point of it in many ways. It really is. It's not the solution to it by any stretch of imagination, but what it can provide is an approximation to it. You're allowing things to change through time, and if you explained it absolutely explicitly and carefully, you would probably find a better description. But perhaps this kind of model might allow you to get most of that immediately and go and solve the problems you want to solve first, rather than having to really, really carefully model it.
Well, thank you once again. Thanks so much.